Friends, welcome to my channel. I am Varsha Gaitonde and this video is about the scientists related to cytogenetics. During the pre-Mendelian period, the first effort in the field of cytogenetics was made in 1590 by Jensen and Jensen. They are popularly called as Jensen brothers, who discovered the operational compound microscope. In the year 1661, Malpighi discovered the Malpighian tubules, that is, capillaries in the lungs of animals. In 1665, Robert Hooke discovered cell by examining the texture of cork by magnifying lens. That means he first observed the cells. In 1672, de Graaf discovered the follicles in human ovary, incorrectly identified them as eggs. In 1677, Leeuwenhoek discovered sperms of some animals but mistook them as wild animalcules. In 1679, Swammerdam developed the Preformation Theory based on studies of the development of insects, a simple enlargement from a minute but preformed animals to adult form. In 1682, Grue was the first implicit mention of the stamens as the male organ of the flower. In 1694, Camerarius gave description of sexual reproduction in plants. In 1707, Linnaeus was called as father of plant taxonomy. In 1761, Colruyter made production of hybrids in Nicotiana, Dianthus, Mithiola, and he gave the equal hereditary contribution of both the parents. In 1809, Lamarck gave Lamarckism or theory of acquired inheritance principle. The theory states that variation in an individual is brought by conscious efforts, reaction to environment, and use of use and disuse organs. The second point says that heredity carries forward the changes that are acquired during the lifetime of an individual. In the year 1824, Amici discovered the pollen tube. In 1828, Brown discovered cell nucleus in the flowering plant Tradescentia. In 1835, Mohl was the creator of modern plant cytology, who first found the cell division and importance of protoplasm. In the year 1838 and 39, Schleiden and Schwann, who were the founders of cell theory, worked independently and coined the cell theory. The Schwann is called as father of modern cytology. In 1841, Remak gave the phenomena of amitosis or direct division. In 1842, Nageli published the first drawing of chromosomes. In 1845, Siebold gave protozoa are the animals consisting of a single cell. Then in 1848, Hofmeister, who illustrated the tetrad formation, identification of two groups of cells at the opposite poles of the embryo sac, but failed to distinguish between the synergids and the eggs. In 1858, Darwin and Wallace put forth the theory of evolution through natural selection. In 1859, Darwin, who wrote a book, Origin of Species, along with that Voyage of Beagle and The Descent of Man. He gave the theory of natural selection. The theory states that variation is constant in nature. Overproduction of offspring brings about struggle for existence. Natural selection operates by elimination of the unfit and survival of the fittest. Then he gave the principle of heredity initially. Then he called hereditary material as gemmules or pangenes. After fertilization, these gemmules separated out to different parts of the body during development. Thus, Darwin attempted physical basis to the Lamarck's idea of inheritance of acquired characters. He also observed the spontaneous changes, which he called as sports. Coming to the post-Mendelian period, Mendelian period is when he started his experiments in peas, and it also includes the publication period. He was active in 1950s till his death 1850s until his death in 1882. 1884, but the period of Mendel's work as well as rediscovery of Mendel's work is considered as post-Mendelian period here. In 1865, Mendel gave the law of heredity like segregation and independent assortment. In the year 1866, Darwin gave the theory of pangenesis, that is a mechanism of heredity. In 1871, Miescher isolated the nucleic acid and nucleoprotein. In 1872, Hartwig gave fertilization as the union of sperm and egg nuclei, that is a general statement. In 1879, Fleming discovered mitosis. In 1879 only, Strasburger gave description of megasporogenesis in plants. He discovered the actual process of syngamy, then fertilization in angiosperms. In the year 1884, Strasburger observed reduced chromosome number during gametogenesis. In 1879, Schleicher gave indirect division, which is called as karyokinesis term. In the year 1880, Hammerstein coined the term protoplast, followed the sequence of early cell divisions in the development of embryos. In 1881, Fouquet gave Xenia effect. Xenia means the pollen produced a visible influence on the hereditary character of those parts of the ovule that surrounding the embryo. In the year 1882, Fleming discovered tiny threads inside salamander, later named it as chromosomes. He also found the mitosis and chromatin terms. In the year 1890, Waldier gave the fundamental fact in mitosis is the formation of nuclear filaments named chromosomes. In 1891, Hanking, who gave X body, which was later named as X chromosome or sex chromosomes, now it is termed as exomechanism of sex determination, means Hanking in 1891 was the first person to contribute for the sex determination process. In 1892, Hartwig gave protoplasm theory, that is proto means first and plasma means formation. In 1896, Wilson, who published the book that is Cell in Development and Inheritance. In 1898, Navashin discovered double fertilization in angiosperms. In the year 1900, that was a major breakthrough in the science when the three scientists independently contributed for the rediscovery of Mendelian principles, and they were Correns, Cermak and de Ries. Correns from German, who worked on hybridization experiments in maize, peas and beans, Cermak from Austria, who worked with peas, and de Ries, who was from Holland, who worked with the evening primrose, that is Oenothera lamarckiana, who coined the term mutation. In the year 1901, he gave mutation theory. In the same year 1900, Gill gave parthenogenesis. Then in 1901, McClung gave sex determination is related to the chromosomes. In 1902 and 3, Sutton and Boveri gave the principle or theory, that is chromosomal theory of inheritance, who worked independently. They gave parallelism between gene and chromosome, means the theory chromosomal theory of inheritance states the parallelism between gene and chromosomes. According to it, the points are: during inheritance, the gene and chromosome both behave like individual unit. Each pair of chromosome can be seen to be different from each other pair. Then each chromosome has individuality. Both occur in pairs, that is, during inheritance, each allele or chromosome occur in pair and contributed from each parent. Each gamete contains only one member of each pair of chromosome. Similarly, each gamete contains one member of each pair of alleles of a gene. Segregation of one allele occurs from its pair and each allele enters in separate gametes. Similarly, each chromosome separates from its homologous pair during meiosis and is included in a separate gamete. In the year 1903, Sutton was called as father of cytogenetics because of his theory of inheritance principles, and in 1904, Montgomery, who gave autosomes which are alike in male and females. In the year 1905, Johansen coined the term gene, genotype, phenotype, pure line. In 1905, Wilson proved the importance of X chromosome in sex determination, who worked with Protoner. And in 1909, he coined the term Y chromosome. From 1905 to 1908, Bateson and Punnett, who discovered the first linkage. Then in 1906, Doncaster and Rayner gave the sex-linked inheritance or found sex-linked inheritance in current mouth, that is Abrascus graculariata. In 1906, Montgomery gave distinction, who made distinction between heterochromosomes and autosomes. In 1907, Bateson coined the term genetics. In 1906, then allelomorph F1, F2 in 1902 and homozygote and heterozygote in 1902. Along with that, he coined epistasis in 1907. In 1909, Jensen gave chiasma type theory of crossing over. 1910, Morgan and co-workers gave sex linkage and genes are located on chromosome, as they proved. In 1912, Morgan made linkage maps of Drosophila. In 1912, Bohr found sex-linked genes in Selline alba, that is Melantium album. In 1913, Sturtevant gave the linear arrangement of genes, or he found the linear arrangements of genes on chromosome or Drosophila chromosomes. In 1914, Belling found the semi-sterility process, the basis of segmental interchange between the non-homologous chromosomes. In 1913 to 1916, Bridges coined the non-disjunction, which is a proof of chromosomal theory of inheritance. In 1917, he found the direct proof that genes are present in chromosomes. In 1919, he coined the term deficiency and duplications in Drosophila melanogaster. In 1916, Winkler found tetraploids, or he worked with the tetraploids in Solanum nigrum from shoots arising from somatic tissues. From 1913 to 1917, Carothers, who correlated the Mendelian segregation and distribution of heteromorphic homologs in meiosis. In 1917 and 1919, Bridges, who recognized the deficiency, duplication, and translocation by genetic tests. In 1920, he gave the genic balance theory, and in 1923 concluded that every individual has its genotype, both male and female potentialities. In 1921, Blaxley discovered trisomics in the Turastomanium. In the same year, Sturtevant recognized that an inversion by genetic test. In 1922, Morgan gave attached X chromosomes in Drosophila. In 1924, Fuelgen and Rosenbach gave specific strain for DNA, that is popularly known as Fuelgen strain. On gentle hydrolysis, the pentose sugar, that is deoxyribose of DNA, liberates aldehyde, which reacts with the basic fission and gives purplish color. In the year 1925, Morgan published The Theory of the Gene, that is a book, and which is very popular. In 1925, Anderson, who gave the proof that crossing over occurs at 4-strand stage through the use of attached X chromosome in Drosophila. In 1926, Kihara and Ono introduced the term auto and allopolyploidy. In 1927, Longley discovered B chromosomes in maize. In the same year, Muller gave X-ray cause on gene mutation. In the year 1928, Hayes made distinction between euchromatin and heterochromatin. In the same year, in 1928, Griffith gave transformation principle. And in 1928, only Gowan discovered genes that affect meiosis. In 1929, Darlington coined the term chiasma terminalization. In the year 1930, Kihara established genome analysis for detecting diploid ancestors of polyploid species, example in wheat. The genome analysis involves crossing between polyploids and diploids and study of chromosome pairing during meiosis in the hybrids. In the year 1931, Crichton and McClintock gave cytological proof of crossing over in maize. In the same year, Stern gave the cytological proof of crossing over in Drosophila. In 1931, Belling gave classical model of crossing over. His hypothesis is like this: homologous unduplicated chromosomes are coiled together. A replication of chromosomes occur first. The replication of interchromatic region occurs, and during the synthesis of interchromatic region, new connections occur between the chromomere of one chromosome and the chromomere of its homologue. In 1932, Darlington published book that is Recent Advances in Cytology. He gave precocity theory of crossing over, according to which the meiotic process starts preconsciously before the chromosomes are in duplicate state, that is, afterwards the theory became invalid. But in 1932, Knoll and Ruska gave electron microscope with a condensed lens. Then in 1931, he discovered NOR, that is nucleolar organizer region. In the year 1933, he along with Bohr discovered the giant chromosome in dipteran insects. In 1933, Painter discovered salivary gland chromosomes in Drosophila melanogaster. These are polytene chromosomes. Then in 1934, McClintock recognized the nucleolus organizer region in maize. In 1936, Casperson developed ultraviolet photomicrography for the study of nucleic acid within nucleus. In the year 1936, Stern discovered mitotic crossing over, that is somatic crossing over. In 1936 only, Sturtevant gave the preferential segregation in Drosophila. In the same year, Sturtevant along with Beadle gave the relation of inversions to crossing over and disjunction. Blaxley and Avery in 1937 gave chromosome doubling by colchicine. Then Sachs in 1937 began the X-ray cytology and cytogenetics. Thom and Steenberg in 1939 made induction of mutations by nitrous acid. Beadle and Tatum in 1941 developed one gene one enzyme concept. In 1944, Avery, McLeod, McCarthy found that DNA is the hereditary material, not the protein. In 1946, Lederberg and Tatum gave the genetic exchange in bacteria. Then Orbach and Robson in 1947 introduced that mutations by nitrogen and sulfur mustard. In 1949, Delbruck and Barley they gave genetic exchange in bacteriophage. In the year 1949 only, Barr and Boutron found the Barr bodies. In 1950, Tijo and Levin used 8-hydroxyquinone for pre-treatment of chromosomes. In 1950, McClintock discovered ACDS system in maize, that is activator dissociator kind of transposons. In 1951, Howarth and Leopold they gave one gene one enzyme hypothesis. In 1952, Chargaff discovered AT-GC ratio in DNA. In the same year, Franklin gave X-ray diffraction data of DNA. And in 1953, Watson and Crick announced the double helix structure of DNA. In 1955, Benzer gave first fine structure of genetic region in bacteriophage. In 1956, Tijo and Levin determined the accurate chromosome number of humans, that is 2n equal to 46. In the same year, Sears developed cytological techniques to transfer genes for leaf rust resistance from wild grass Aegilops umbellata to bread wheat. Aegilops umbellata has the basic chromosome number of 7 and the haploid number of 14, and bread wheat is 46 chromosome number. In the year 1960, Barksky gave somatic cell fusion. In 1957, Benzer classified gene into cistron, recon and muton, and the same person in 1961 found the mutation hotspots. In 1961, Driscoll and Endelberg discovered E. coli F factor, which is a DNA plasmid. Then in 1961, Lyon-made Lyon's hypothesis or single active X hypothesis. Single Y chromosome or X chromosome in male and one of the two X chromosomes of females are active like autosomes, but one X chromosome of the female is inactivated by the process called heterochromatization that occurs early in the embryonic development. In the year 1962, Riss and Plot discovered chloroplast chromosome. In the same year, Silver and Ozeki discovered cholinogenic factor, that is Col factor, as a DNA plasmid. In the same year, Arber first gave the first evidence for the existence of DNA restriction enzymes. In 1963, Nass and Nass discovered mitochondrial chromosomes. In the same year, Marmor made DNA hybridization in vitro. In 1964, Britton and Warning discovered repetitious DNA in eukaryotes. In 1965, Duprat discovered the whole mount electron microscopy. In 1968, Arber, Smith and Nathans gave restriction and modification of enzymes. 1968 to 70, Kasperson and colleagues worked for the development of fluorescent and GM staining for eukaryotic chromosomes. In 1969, Jonathan and co-workers isolated the first gene. In 1970, Pord and Gall made DNA or RNA hybridization in vitro. In 1972, Khorana and co-workers synthesized the first artificial gene. In the same year, Boyer's research group, that is Berg and colleagues, produced the recombinant DNA. In the year 1972 and 73, Stanley Cohen and Chang gave the technique of DNA cloning. In the year 1972, Singer and Nicholson gave fluid mosaic model, that is the inflow and outflow through cell membrane, which was a two-dimensional model. In the year 1974, Thomas Jr. and Wilson discovered widespread occurrence of the hairpin-like structure, that is palindrome, resulting from inverted repetitious DNA located at intervals along the chromatids of eukaryotic chromosomes. In 1975, Hargovinda Khorana made artificial synthesis of a gene. In 1977 to 78, Sanger and co-workers discovered overlapping genes, including the interrupted genes. Means they were not the persons who exactly found the interrupted or split genes, but after that Phillips Sharp gave contribution for the split genes. In 1981, Anderson gave first mitochondrial DNA sequence. In 1983, Evans and co-workers discovered cyclins. In the same year, Murray and Zostak made first synthetic chromosome. In 1983, Bender and co-workers gave the concept chromosome walking. In 1987, Grieder and Blackburn discovered telomerase, that is needed for the synthesis of telomere. In the same year, David Burke, Corley and Olson developed yeast artificial chromosome. In 1989, Dretta and co-workers gave the role of cyclins in M-phase kinase. Coming to the genetic engineering period, in 1973, Cohen and Boyer, along with other co-workers, gave the insertion of a gene and DNA cloning. In 1976, means Cohen and Boyer gave the systematic explanation to DNA cloning. In 1976, GeneTech was an company which established the first genetic engineering company. Means Genetic was the first genetic engineering company. In 77-78, Sanger and Coulson gave rapid sequence of DNA. Along with Maxam and Gilbert, they gave the methods of DNA sequencing. In 1983, Gusella and co-workers found the location of Huntington's disease on human chromosome 4. In 1984, Jeffrey developed the genetic fingerprinting. In 1986, initiation of the Human Genome Project. 87, production of the first comprehensive human genetic map. In 1988, creation of National Center for Human Genome Research. In 1990, launch of Human Genome Project in the therapy on 4-year-old girl with an immune disorder known as ADA deficiency, but procedure did not work. Mary Claire King demonstrated that the gene on chromosome 17 causes an inherited form of breast cancer and increased the risk of ovarian cancer. In 1992, production of a rough human genetic map by international collaboration. In 1995, Institute for Genome Research put forth the sequencing of different organisms including E. coli and all. The same year, publication of first complete DNA sequence of the genome, that is a free living organism, Haemophilus influenza, which is a bacteria. In the same year, Monsanto company developed the Roundup Ready soybean. In 1996, international collaboration involving 600 scientists from Europe, North America and Japan gave the first complete genome sequencing of yeast, that is Saccharomyces cerevisiae. In 1996, transgenic maize, potato, cotton expressing Bt toxin grown in the United States on a large scale. In 1998, first genome of the multicellular organism, that is 97 megabase DNA sequence of roundworm. In the year 2000, Celera Genomics made the first draft of sequence of 90% of the human genome. In the year 2000, Institute of Genomic Research completed Arabidopsis genome map. In 2001, Syngenta and Merit Genetics completed rice genomic maps. Then in 2002, Syngenta completed Japonica rice genome sequence. In the same year, Beijing Genomic Institute completed Indica rice genomic sequencing. 2004 onwards, many of the genomes were sequenced, means the many organism genomes sequenced and resequenced, and that became a good work for development as well as identifying the good genes for different traits which human were interested. I hope this information is helpful for you. If you like this video, please do not forget to hit the like button. Please subscribe my channel. Keep on liking, commenting, sharing. Thank you once again.